Hello, I'm a student from Harmony School of Innovation, Katie, and this is our project, gBlock Lyme disease RNA vaccine of outer surface protein A.
An RNA vaccine for Lyme disease is vital for immunization as 14% of the world is affected by this illness, yet there is currently no CDC-approved vaccine aimed to cure it. We hope to reduce the number of cases through eventual immunization. While working on our project, we did thorough research on previous effective vaccines to ensure the conciseness of our gBlock. Some limitations of developing this vaccine include faulty gene transcription, deterioration, and sterility issues. By taking the initiative to research and test an RNA vaccine, we hope to contribute to the field of Lyme disease interventions. For some background information, Lyme disease or Borrelia burgdorferi is a flu-like disease which causes symptoms such as fatigue, headaches, bullseye rashes, soreness, and fevers. If left untreated, this infection can spread to the cardiovascular and nervous systems. However, it can be diagnosed through symptom screenings and ELISA testing. Borrelia burgdorferi is carried by infected ticks and spreads when they bite and attach to a host. For our RNA vaccine, we targeted the outer surface protein on Borrelia burgdorferi to eradicate the disease. When synthesizing our RNA vaccine, we chose to use a T7 promoter to initialize transcription. T7 promoters have previously been used in successful mRNA vaccines such as the SARS-CoV-2 vaccine. Furthermore, they are short yet effective, making them cost-efficient to produce on a larger scale. We also chose to use the human hemoglobin beta subunit gene for our untranslated region because it has been proven to regulate high levels of protein expression in the body, allowing our vaccine to be expressed adequately. We also used a Kozak sequence to initiate translation in our gene design. These components must be used in vaccines to ensure the targeting of the OspA and prompt the body to produce the necessary antibodies to fight it. We hypothesized that if we synthesize, transcribe, assess, and purify our gBlock, it will produce significant yields of intact RNA which could be capable of eradicating Borrelia burgdorferi.
For our procedure, we began by designing our DNA sequence. We started by adding a T7 promoter to initialize transcription, then added a 5' UTR derived from the human hemoglobin beta gene to regulate high levels of protein expression. Next, we added a Kozak sequence to initialize translation, and then added a start codon. We then added a DNA sequence which is derived from outer surface protein A's protein sequence, then optimized for the human body. We then added a stop codon and then a 3' UTR which is also derived from the human hemoglobin beta gene. We ordered our designed DNA as a gBlock, which is what the company we ordered our synthesized DNA from calls synthesized DNA. Once we received our sample, we diluted it to 20 nanograms of DNA per microliter of RNAs and DNase free water, and then heated it for 20 minutes at 50 degrees Celsius, then stored it at -20 degrees Celsius until we were ready for RNA transcription. For RNA transcription, we took 8 microliters of our diluted DNA, which we briefly heated, and then added 2 microliters each of ATP, CTP, GTP, PUTP, 10x reaction buffer, and T7 RNA polymerase. We then centrifuged our sample briefly, then put it in a thermocycler for the 3 phases of PCR, and then set it to 37 degrees for overnight transcription. The next day, we used a nanodrop spectrophotometer to quantify the amount of RNA we transcribed by pipetting 1 microliter of our sample to the pedestal and setting the free water as a blank. We then quantified our RNA. To determine our RNA's integrity, we ran a gel electrophoresis for 2 minutes by adding 2.5 microliters of our sample into the wells and running it at a power supply of 100 watts. We visualized whether our RNA was intact.
After gel electrophoresis, we needed to make sure we had pure RNA, getting rid of excess buffers, proteins, and nucleotides. For our procedure, we used the lithium chloride purification method, as is shown to be highly effective at purifying RNA. We first added 1 microliter of turbo-DNase to our solution and incubated it for 30 minutes. This ensured we got rid of our template DNA strand. We then stopped the reaction and added 30 microliters of nuclease-free water and 30 microliters of lithium chloride precipitation solution. We incubated this for 30 minutes and centrifuged it. This ensured we got rid of our proteins and buffer solution. We got rid of the supernatant and we centrifuged the solution again with 70% ethanol alcohol. This ensured we got rid of unincorporated nucleotides. After going through our whole procedure, we were able to quantify that we had 7,563 nanograms of RNA generated after testing 1 microliter of solution on the nanodrop. Since we had 20 microliters of solution, we were able to calculate that we had 151,270 nanograms of RNA. This yield holds significance as it surpasses the average RNA dose required for a vaccine, which is about 30 micrograms per 0.3 milliliters of solution by substantial margin. Using the same ideals, we would have 2,269.05 micrograms of RNA per vaccine dose. This ensures we have an ample supply even in the event of degradation during future procedures. Moreover, the methodology yielded intact RNA, evidenced by the absence of smearing and gel electrophoresis bands, and the observation of a cloudy precipitate confirms a successful RNA purification. Hence, we were able to conclude that our method of transcription and purification accumulated a substantial yield of intact RNA, which promises implications for future research.
For the ELA component of our project, we wrote a narrative essay about our journey to realizing the importance of a Lyme disease vaccine by recognizing that different people's situations may prevent them from getting Lyme disease treatment. You may pause to read now. For the social studies component of our project, we wrote about how Lyme disease is observed in the world. You may pause to read now. Thank you for watching HSI Katie.